Okay, I'm going to get started then. Welcome everyone to a review session for Exam 1. And I have some questions that people posted through our exit survey this week, but since you guys are already logged in and participating, I'm going to open it up to questions right away that you guys have.
Just going through the study guide here, in terms of the long essays with Pasteur's SNAC experiments and the key conclusions about microbes and biogenesis. In terms of what you're looking for the biogenesis, are you looking for to talk about the spontaneous generation versus biogenesis, or
Yes.
what, can you clarify a little bit on?
Yeah, absolutely. It's similar to what we read about in Chapter 1, as well as the Chapter 1 homework that asked you to explain the data and how it either was supporting or refuting the idea of the prevalent idea of that time of spontaneous generation. And so you need to know the difference between spontaneous generation and biogenesis, as well as this key set of experiments that Pasteur did, that what he did and what the results mean. So the conclusions he drew and how they supported the idea of biogenesis, where life arises from pre-existing life, and refuted the idea of spontaneous generation, that if you have the conditions for life to exist, it won't just appear. There has to be some pre-existing life there. Does that answer your question?
Yes, I think so.
Okay.
Is this like the homework where you're really looking for keywords like spontaneous generation and biogenesis specifically, or are there any kind of key other focused areas we can?
In general,
talk about?
Yeah, in general, for the open response, I'm looking for you to give an answer and back that answer up with explanation. So I'll ask a question, but if you just give me the answer without explaining it, you won't get full credit. And I'm also expecting you to write in full sentences to get full credit. So I generally have a series of maybe 4 or 5 items that I'm looking for. Often it's a keyword, definition of that term, or using it properly. For things like Pasteur's experiments, it would be explaining the conclusions as well and understanding that, as well as what he did. Yeah. Mm-hmm.
Okay.
Anyone else have a question? Ulrich?
Hi, Professor Olkin.
Oh, hi. Who's this?
Hi, sorry, this is Siobhan Claflin. I'm on the phone, and the WebEx wasn't working for me. I apologize.
Oh, no problem.
Could you discuss one of the study points on the guide was the relationship between infection and disease and virulence and pathogenesis?
Okay, so these are 4 key epidemiologic terms that I'm asking you to understand and to be able to distinguish. So let's just start. So they can be paired with infection and disease can be paired, and virulence and pathogenicity can be paired. So let's just start with infection and disease. So you can actually be infected with a pathogen but not get disease from the pathogen. So that's basically how they're distinguished. So for example, if I had the flu last year and I get infected with the same virus this year, I will not have the flu because I already had the flu and my body has built up immunity to that. So I'm no longer susceptible to the disease, but I can still get exposed to the flu. The flu comes around every winter season, right? So it's not that my body somehow miraculously prevents the flu from entering my respiratory tract, the virus that causes the flu. But that microbe, once it gets into me, even if it gets into and infects the cells of my respiratory tract, which is the host cell for this virus in the upper respiratory tract, even if that happens, I won't get sick from it because my secondary immune response has memory to it, so I won't get the disease. So that's an example illustrating the difference between infection and disease. So you can have both infection followed by disease, or you can have infection without disease.
If you are a carrier, are you infected?
Yeah.
Okay.
Yeah, so basically infection is the microbe or the pathogen is in your body. In the case of bacteria and things like that, so you're carrying it. So you know, in the example of cholera, people can carry the bacteria that causes cholera without actually being sick with diarrhea, and they can spread it. But obviously, if they're sick with diarrhea, they're having the disease of cholera, and they're spreading it a lot more than a carrier would. Yeah.
Okay.
Or like with typhoid Mary, I think was an example given in your book. She was a carrier. She had no symptoms. She said, I'm not sick. I can still serve food to these people. I might make food for these people. But really, she was spreading typhoid by that job that she had. And she was banned from it, from having that job after they realized she was the source of infection.
Okay.
Okay, and then for the other two terms, virulence and pathogenicity. So while they're closely related, they're not the same. So virulence is a measure of how severe a pathogen can, how severe a disease resulting from a pathogen can be. So there are actually factors that are genetically determined that pathogens have that non-pathogens will not have, that distinguish them, and we call them virulence factors. And so it's a measure of severity. We can measure it in as far as the lethal dose or the infectious dose of a pathogen that's necessary to make 50% of the population sick or to kill 50% of the population. But pathogenicity is the process of causing disease that pathogens have. Okay?
Yep. Thank you.
Sure. Any other questions?
I have a question.
Sure.
So for the very last question,
Mm-hmm.
could you give us some tips about those?
Okay, so that's distinguishing the features or the question about the open response? Which question are you referring to?
The features.
Okay, so the last bullet point that I have on the first page of the study guide is discuss the features that distinguish members in the microbial world as far as cell structure, reproduction, metabolism. So these are some of the general features that I went over in the lecture for Chapter 1, as well as that were in those introductory parts of the chapters that I asked you to read for Chapter 10 and 11 and 12. Let's just take a moment to look at the figure that I had. Let's see, I think that's here. Yeah, so I gave a figure that showed the classification of members of the microbial world. Remember, there are two major groups of members, those that are living or microorganisms and those that are non-living or microbial agents. And then the microorganisms are further divided into the three domains of life that are used to characterize all life, not just microorganisms, so bacteria, archaea, and eukarya. Two important distinctions that those groups have is in their cell structure. So anything that's non-living, because the cell theory tells us that all life has the same common basic structure of a cell, a lipid bilayer around its contents that separates what's inside the organism from what's outside in the environment. So non-living members of the microbial world don't have that structure. We call them acellular. Remember, we talked about, I I can't remember if it was your section, but we were talking about viruses, how they have genetic information in the form of either DNA or RNA, but they don't always have a membrane around them. Some of them are envelopes, some of them aren't. They all have a protein coat around their genetic information, but that's it. Some viruses, like poliovirus, it's just very simple. Just has a protein coat or capsid that surrounds the genetic information of DNA in that case. Okay, in the case of living organisms that are members of the microbial world, we have microorganisms like bacteria, archaea, fungi, some unicellular algae, protozoa, and those multicellular organisms that don't really fall into the definition of microorganism, but we put them along with all of the other members of the microbial world because they are parasitic and cause infectious disease, and that's the helminths. So the bacteria and archaea have a fundamental difference in their cell structure than the eukarya, and that's what we call prokaryotic cell structure. Pro meaning before there was a true compartment for DNA, and U meaning a true, and karyo referring to the DNA. So prokaryotes don't have a nucleus. They don't have any internal compartments. So they're much simpler in their cell structure. They just have one lipid bilayer that separates them between what's inside the organism from what's outside in the environment. Whereas the eukaryotes are more complex. So you've learned about these in anatomy and physiology because human cells have a eukaryotic cell structure. And that's the same kind of cell structure that you find for microorganisms like fungi, protozoa, unicellular algae, all of these protists that would be considered members of the microbial world. So that's what I'm looking for as far as the cell structural differences. There are also reproductive differences. So in general, prokaryotes divide more simply than eukaryotes. Eukaryotes divide mitosis or some kind of variation on mitosis. Prokaryotes, such as bacteria, divide just kind of a carbon copy of each other. So the one cell goes through a fission process where it divides into two equally sized cells. There's no growth or development associated with that. It's just one cell to two, two to four, four to eight, kind of this exponential growth that you don't always see with eukaryotes. And then, let's see, what else did I put there? So don't worry too much about the metabolism now. There are some differences, but we'll learn more about the differences in the metabolism when we study metabolism of microbes in chapters 6. I think it's chapter 6, 5 or 6, something like that. And then just in general, you should know that algae are not human pathogens, although they can indirectly cause disease. They're not direct primary pathogens. But all the rest, fungi, protozoa, and helminths all have, not all of them, but they include examples of human pathogens. Archaea, again, that's a domain that does not include any human pathogens, but bacteria, of course, it is the major source of human pathogens. As well as viruses, we know, are human pathogens, but not viroids. Remember, viroids are infectious agents of plants, not animals or humans.
Right.
And then prions are examples of human pathogens. Okay, so just know that generally, like, you know, what includes human, which of these groups of the members of the microbial world include human pathogens and which don't. And we'll talk about more specifics as we get into further into the semester and further into the textbook.
Okay.
So hopefully that clarifies that point for you. Do you have any other questions about it?
No, thank you. That was really
Okay.
helpful.
My question sort of relates to that. Sorry, it's Siobhan again.
Okay.
When we're organizing things in the microbial world, I get confused about the infectious agents just because they're non-living, and sort of the definition of a microbe is a living organism that requires a microscope to be seen.
Okay, so
right?
The microorganisms have that definition, and maybe the book has a different definition than I do, but I use microbe as an umbrella term for everything living and non-living that are members of the microbial world, whereas microorganisms I use for the definition that you just gave. Yeah.
Okay.
Yeah. Any other questions for the moment?
In one of the shorter answer ones, you asked about the general steps of the bacterial pathogenesis. Is that just how bacteria turns into a disease, or what? Can you expand more on what you're looking there?
Yeah, right. So pathogenesis is the process of causing disease. So there are basically 4 steps that all pathogens have in order to cause disease, which is basically causing damage to our system. And then we get the signs and symptoms of the disease because of that damage. So first of all, of course, there has to be the entry of the bacterial pathogen into our body. Then it has to attach in order to be able to colonize, which is another important step. Now some microbes, that's it. Once they colonize, they're able to produce factors that would be damaging to our tissues and cause the disease. Others can invade beyond the surface of our tissues. So for example, Shigella causes diarrhea just like E. coli, but E. coli colonizes the gut and causes diarrhea, whereas Shigella invades past the gut epithelia into the underlying tissue and causes its damage in that way. And it causes dysentery, a much more severe diarrheal disease. So these are all steps of bacterial pathogenesis. And one of the most common steps then after colonization is toxin production, although not all bacterial pathogens cause toxin. The ones that produce toxins, but the ones that do tend to cause more damage and more symptoms.
I thought that toxin production was sort of one of the virulence factors. The stuff that I have for pathogenesis is entry, attachment, immune avoidance, and obtaining nutrients from hosts.
Yep, those are all key. So pathogenesis, though, is the process that also includes these virulence factors like toxin production. But certainly those 4, it has to always, whether it invades or not, it has to get food in order to be able to survive in us as a host. It has to attach, otherwise it'll be flushed out, for example, by the movement of our colon or our GI tract before it can cause any damage. So it has to attach, it has to colonize or grow, and in order to grow it needs to acquire those nutrients. So there are certain virulence factors even among even at that stage that are helping to acquire the nutrients it needs. Like iron is a really tough nutrient for microbes to get when they're in our body, because most of the iron that we have is not freely available. It's bound up by our own proteins, kind of hidden away from something like a microorganism. So a very important virulence factor for many pathogens is the ability to sequester the iron to, for example, break open or lyse our red blood cells so that the iron in there can be released for the use of the pathogen. Okay? So, but yes, those 4 steps that you stated, Siobhan, are the correct pathogenesis steps that I'm looking for, the general steps. Yeah.
Okay, cool.
We'll learn more about the immune avoidance part of it later in the semester, but obviously that should be familiar from anatomy and physiology, and we'll go over it in more detail later in the semester. This kind of balance between our body's desire to fight off the infection and the pathogen's desire to persist in our body.
Do I have this correct, sort of a simple definition where the agent is the microbe that is invading the human body, or the animal is the agent? So that would be the...
Yeah, the agent is always the microbe that's causing the disease. So you would never have a host, or you would never have the host animal or the human as an agent. It's always a microbe or a pathogen, really. We're talking about pathogens in the case of agents, either living or non-living.
Okay. Yeah. And then the reservoir is sort of the environment or the location where that pathogen is normally, that harbors it normally, I guess?
Harbors, yeah, so the source. So, for example, some diseases come from contaminated water, like Vibrio cholera bacteria. So we're not normally carrying around these pathogenic bacteria, but once we're exposed to that contaminated water, then it can make us sick, right? So that's the reservoir for that pathogen. But it doesn't have to be outside the body. So I gave you an example of this young woman, Carrie, who had a urinary tract infection that was actually from her normal microbiota. So the human body can also be a reservoir.
Okay.
That's a little confusing because in
in
that example, like, she was both the host and her body was the reservoir. She didn't get it from another woman's genital tract. She got it from her own, right?
Right. Okay.
So, and that's true also for the more common infection of E. coli. E. coli is probably the number 1 cause of urinary tract infections, and that's not from the perineum, but from normal microbiota of our gut, and just like poor hygiene may contribute to that. Did someone have a question in there I missed? Or a comment?
That might have been me. I was just saying that a question I had is whether or not the human body could be a reservoir based off of that example, but you already answered it.
Absolutely, yes. It is quite often. Yeah. And the diseases that have humans as a reservoir, whether it's an infectious disease like the flu, or I guess a better example would be polio, because polio doesn't have like an animal reservoir or environmental reservoir like water or food. So the virus can be eradicated potentially from the human population, and once it's eradicated, so for example the smallpox, the only reservoir were humans for smallpox, and now we don't have smallpox anymore because we successfully eliminated it from anyone in the world. And so that's it, we're safe from smallpox. Right? Yeah, through vaccination, yes. So the WHO is trying to do the same thing with polio. So it's easier to control those diseases. Where something like Zika, you've got mosquitoes. It's really unrealistic to ever expect to eradicate Zika virus or Zika disease from the human population, because there will always be mosquitoes harboring it, and you really can't eliminate the world population of mosquitoes, although there are some strategies for control that try to do that. So yeah.
So can humans be vectors?
No, vectors by definition are not organisms well, they're carriers, so they're more, for example, from the insect or the like mites or lice or things. They're not humans. They can be responsible for transmitting a disease, but we don't use humans as we don't use the term vector for humans.
Okay.
Yeah.
If I could go back to the relationship between the agent, reservoir, host, and infectious cycle of disease, when you're talking about the infectious cycle of disease, do you mean the portals of entry and exit, or can you expand upon that a little bit?
Okay.
Just in terms of the infectious cycle of disease part?
Yeah, let me see. Which point is that?
It's describe the relationship between
Oh,
agent,
agent,
reservoir.
reservoir, infectious cycle of disease. Okay, yes. So,
Yeah, I'm just trying to figure out how the cycle fits in there.
right. So, basically, the cycle is how the disease is transmitted. When I'm
Okay.
talking about that cycle, so the persistence of an infectious disease relies on the agent being present, either in a host or well, being present in the environment, either as a human reservoir or an environmental reservoir or some other animal reservoir. So I think I have an image, yeah, here. Let's look at this a little more closely. So I like to think of it as a triangle relationship where they're all tied into each other. Let's see if I can get this to be.
Is that the triangle
Okay,
that says agent, host, environment, and then disease is in the triangle?
Yep.
Okay.
Right, so to explain a disease, there are these three factors that always keep the disease present in the human population. And you have to have all three of these in order for it to be present in the human population. So there's always a cycle. The cycle is that the environment, or you can also label that the reservoir. It can be environment or human or an animal. So that the presence of the agent in the reservoir, and then that transmission of that agent to a human host, since we're focusing on human diseases here, but it could be an animal host in case of animal disease, that has to be, those elements have to be present, right? So if there are no susceptible hosts available in the human population, then the disease will die out. It will not persist in the human population. So all of those elements have to be present in order for an infectious disease to occur in the human population. So that's really the cycle, of which I'm really referring to transmission when I talk about the cycle.
Okay, thank you.
Mm-hmm. If anyone has any other questions, just speak up. But there were a few questions that I just want to go over quickly um, from the exit survey that I posted that was due last night. So someone was asking to distinguish between pandemic and epidemic. And these are epidemiologic terms that you should be familiar with. Pandemic, let's see which chapter, no, not that one. So pandemic means they're referring to the presence of disease, but pandemic means that the disease has spread worldwide. So remember back to 2009 when we had an influenza pandemic where it spread originally, I think one of the first cases was in Mexico, but it spread to all the rest of the continents and countries in the world. And so that H1N1 flu is an example of pandemic. Also, HIV is a pandemic. You know, it's pretty much anywhere in the world you'll find cases where people are HIV positive. An epidemic, though, on the other hand, is more geographically localized. So you might have an epidemic without a pandemic. Polio can be epidemic in a few countries, like I think Nigeria still has polio epidemics. But for the most of the rest of the world, we're protected from it by vaccination. And endemic means that it's there from year to year. There's not just kind of one large number of cases. So actually the Zika virus could be an example of an epidemic. People were worried about it spreading to the world and becoming a pandemic, but it didn't. Let's see. Do I have it? I don't remember where I put it in the... Yeah, here it is. Right. So those three terms of endemic, epidemic, and pandemic, you should know the difference between them. So certain areas in the world harbor, for example, malaria. We don't have malaria in the U.S., but other parts of the world, it's endemic. Cambodia, for example, it's endemic. They always have it. They always have it each year. There are cases of people getting it each year. Another question that I had from the exit survey was, do we need to know all the detail about the different kinds of microscopes? Just asking you to be able to distinguish between the features of a light microscope like we use in lab and electron microscope as far as like which microbes we can view with which and which are more limiting in our ability to see microbes. Not really any more detail than the features of, you know, some offer greater magnification and greater resolution than other types of microscopy.
I have a question. I'm really
Sure.
uncomfortable with the metric system. I have a really hard time. Do you have any resources? I've been to Khan Academy and different, you know, friends have given me different ways, like you make up a word for the kilo, hectodeca, base, deci, centi, milli. But do you know of any resources where I could just maybe get more comfortable with it?
I think practice. I think you've probably done the right thing of looking at the ways that other people explain it, and you can narrow your learning to just the micro scale of, you know, we're not going to talk about kilometers, we're going to talk about nanometers or
Right.
millimeters, or I'm going to ask you about micrometers, but nothing bigger than a millimeter. But you
So if
should know the
millimeter,
relation.
sorry to interrupt. Millimeter
Um,
is, if
mhm.
that's 1/1000, then nanometer would be 1 in
Yes.
10,000?
So actually it goes from meter, and then milli is 1/1000th of a meter,
Right.
and then 1/1000th of a millimeter is a micrometer, and then
Micro. That's right. Then the nanometer.
then the
Got it.
nanometer. So it's
Got it.
actually
Okay.
10 to the negative ninth,
See, that
right? 10 to
confuses
the
me when
negative
it's that.
ninth meters, or uh, a zero with a decimal point, and then you have 8 zeros and a 1. Okay, so
Okay.
point 0.000000001. So
So
is
when they say to the something power, that's actually the number of decimal points from the, or the number of
yes, right. The
spaces
number of
before.
times
Okay.
that you move the decimal point over before
Right.
you put, if it's 1
I just have to
nanometer before you
practice.
put the number 1, or if it's 5 nanometers
Okay.
before you put the number 5. And yeah, so
All
it's
right.
it's I mean that's the nice thing about the metric units. It's not like 12 inches to a foot. It's just
Yes.
tenths. So you're basically just
Right.
moving the decimal one way or the other, but you have to remember that, just try to get it straight in your head that nanometers are really small. So if you're talking about a
Yeah.
meter, they're not 0.0000001 nanometers in a meter. It's the other way around, right? There are one with 8 zeros after it, one with one with 9 zeros after it. Um, so not 100,000, not 100 million, but yeah, 1 million um, uh, is, what did I say, a thousandth, a millionth, and a hundredth. I have to write it out myself. It's
Yes.
one with 1, 2, 3, 4, 5, 6. It's 9 zeros, so it's not
Okay.
1,000. It's not 100,000. It's 1 million, 100 million, 1 billion,
Yeah.
right?
Oh, it all makes
So
sense now.
1 billion nanometers. You have a lot of nanometers in a meter, not the other
Okay.
way
Yeah. Yeah.
around.
Yeah.
So
Right.
just
Right.
remember that if you start writing a decimal for a nanometer, it's probably wrong. The nanometer number
Okay.
should be big when you're converting from meters, and the meter
Okay.
number should be small when converting from a nanometer.
Right.
Right.
Okay.
So
Good.
that there
Good
are
clue.
some
Thanks.
tricks to, and if you just, I'm not going to ask you in between a nanometer and a micrometer. Just remember that they're 1,000 times, they jump by 1,000
Okay.
times. Yeah.
Okay. Thank you.
Yeah. Okay. If there are no other questions, then I will just wrap up, and if you think of something later, when you're studying tonight or tomorrow, then just post it to our website. Remember, there's at the top of the website, just before week 1, I have a discussion forum that's called Ask Professor Olkin here. And you can just post a question there, and I'll post the answer. Or if it's something more personal question, then you can email me directly. Okay?
Okay, great. Thanks for your time.
Okay.
Yes, thank you.
Yeah, thank you.
Okay.